Hello and welcome to the 8th session of Brain Matters. Today we'll be discussing emerging rhythmic patterns and their analysis. I'm very pleased and honored to moderate this session. I'll give an opening about the HBP's goals and my work within them, which will be followed by 3 presentations from our delightful speakers and a Q&A session beginning at 5:15. So during the course of the presentations, I encourage you to post your questions in the Q&A box at the bottom of your screen. And we will address them all at the end of the presentations. So my name is Jennifer Goldman. I work as a postdoctoral fellow in the laboratory of Alain Destex at the EITN. And we study methods to try to bridge scales between microscopic and macroscopic dynamics in the brain. So my talk is called A Scale Integrated Approach to Brain Dynamics, from Single Neuron Biophysics to Macroscopic Neural Dynamics. This research was conducted in the lab of Professor Alain Destex and has become a part of the Human Brain Project, specifically Showcase 3 and in eBrains. So we were inspired at first by states of matter, where we know that there are macroscopic behaviors that emerge like solids, liquids, and gases that are dependent on microscopic phenomena, where the microscopic interactions are stronger or weaker, gives rise to the emergence of these macroscopic properties of the matter. And so it is with brain states, where an individual might be in a deep sleep, an awake but restful state, or an awake active state. We can record macroscopic signals from the brain that vary in the complexity and the magnitude of these recordings. If we look at the mesoscopic scale, for example, with local field potentials, or the microscopic scale, for example, with spikes, we can see that underlying these slow waves, there are periods of synchronous firing between the cells, the excitatory and inhibitory neurons, and periods of silence. So these periods of synchrony, synchronous firing and silence give rise to these slow waves. Now, we believe this might be in analogy to states of matter, actually because changes in the interactions between the neural populations that lead to these large, simple waves that you see in unconscious brain states, or the small complex signals that we see in active, especially active waking states. So we know biophysically that this transition between slow waves to this asynchronous awake-like dynamics can be occasioned by the increase of spike frequency adaptation. So essentially what happens is when cells are in this synchronous firing state, that they will build up a spike frequency adaptation that self-inhibits. This is due to potassium leak channels for the most part, which self-inhibits the cells, driving them into a down state, at which point the self-inhibition wears off and the cells are able to transition into an up state again. So the addition of neuromodulators, including acetylcholine, cause the closing of these leak channels in the membrane, blocking the spike frequency adaptation. So in order to model this phenomena, students working previously in the lab of Alain Destex, including Jan Zerlo and Matteo Di Volo, developed models of populations of spiking neurons, these adaptive exponential neurons, which you can see, depending on the spike frequency adaptation parameter, go from this asynchronous state to this synchronous on and off periods. So further work developed these models into mean field models of population dynamics that are able to capture, instead of using thousands of differential equations, using only a few differential equations, and thereby making the computations much more tractable. So when I joined the project, I began working very hard with Lionel Kusch in supervision of Victor Yerza to use these ADEX mean fields developed by Alain and his previous trainees to couple together by the human connectome. Now, we used the weights and the tract lengths to inform the connectivity strengths and the delays in these models. thereby producing whole brain models that are informed by the microscopic biophysics of neurons. So what you can see in this model of approximately 70 brain regions, each brain region represented by a mean field model, that in low adaptation conditions, we have this asynchronous, small magnitude, very complex signals. And the more we increase the spike frequency adaptation, the more we get synchrony and slow waves, reminiscent of slow wave sleep and in deep anesthesia. So what we can say is that increasing spike frequency adaptation to simulate acetylcholine availability at the microscopic scale results in the emergence of these conscious and unconscious like global brain dynamics. So in collaboration with two very gifted students, Changyan Nyam and Bahar Hazal-Yasin-Kaya, we managed to replicate these signals with the whole brain model and showed that the power spectrum, which is often used to quantify different brain states, has a peak of the power spectrum that is closer to the alpha range in the wake-like condition, whereas the maximum of the power spectrum is much lower in the frequency domain in the sleep-like case, very reminiscent of the data. So further, we can look at the coupling between the nodes and show that in fact, when the spike frequency adaptation is low, producing a wake-like state, we are able to replicate empirical data showing that the correlations between different brain regions are much lower. As well, we can see that the phase coupling between the different nodes is much lower than when we actually increase the spike frequency adaptation in the sleep-like case. So we can say that macroscopic coupling can emerge from microscopic modulatory processes. So this is all attempting to simulate, reproduce spontaneous behavior of the system. And so one way to extend this analogy would be to say that in fact, the spontaneous state of the system gives some information about how it will respond to a perturbation. So we might think of the wake-like state and this complex dynamics as being more gas-like, whereas the sort of more resting or unconscious brain states may be more like liquids or solids. So previous work from Marcello Massimini and his colleagues have shown that in fact, when you perturb the brain, when it's awake, the signals are smaller in magnitude, but they propagate much further in space and time. Whereas when subjects are unconscious, there's a large magnitude response that stays very proximal in space and time. So this has been quantified with the perturbational complexity index. So in waking subjects, you see a high level of the PCI, whereas when the same subjects are in deep sleep or are anesthetized, there's a much lower value of the perturbational complexity index. So finally, we were able to replicate this in our multi-scale human brain model, showing that in awake-like states, these asynchronous states, you have a small magnitude response, but which propagates very far in space and time. Whereas when the model is in a sleep-like condition, you have this local large responsiveness that doesn't propagate very far. And this is quantified here. So we can say that the spontaneous behavior of the system modulates its responsiveness to perturbations. And you can see all of this work described in a recent preprint from our lab. And I'd like to end with this thought that slow waves might represent a kind of universality class. And it's something maybe we can discuss at the end of the seminar, there are various ways to put the brain in a slow wave state. For example, head trauma, neurovascular disease, anesthesia, and neurodegenerative disorders all result in prominent slow wave synchronous activity. So our future goals using this TVB-ADX model I've described to you is to study cases of head trauma, anesthesia, neurodegenerative disorders, and neurovascular disease, able to modify the parameters in the model that would simulate these processes. Next, we'd like to personalize the models with the individual subject's anatomy, their physiology, genetics, that are all informed by longitudinal data capable of predicting fluctuations and transitions over time. And this is all in the global context of the Human Brain Project, what we're all working toward accomplishing. So this is the end of my presentation. Thank you, all of you. You can find information about this model and all of the work that's being produced by the Human Brain Project in the knowledge graph, and also interact with much of the code and the data in the eBrains lab, which you can see here. Thank you to all my wonderful supervisors and collaborators. And that's all I have to show you for now. So to stay well on time, I will introduce our first speaker, who is Antonio Pazienti. So thank you for any questions you have about my presentation or the presentations to follow. We will address them during the Q&A period. So Antonio has his diploma in physics from Sapienza University in Rome, with a focus on dynamical systems and neural networks. He did his PhD in physics at Potsdam University in Germany, where his work was carried out in the neurobiology department of Free University in Berlin, at the Reichen Brain Institute in Japan, during which time he was a member of the Bernstein Center for Computational Neuroscience. He's conducted his postdoc in the European Brain Research Institute in Rome, where he's learned electrophysiology, including recordings in dynamic clamp. He is a researcher at the Italian National Institute of Health, interested in the cellular and network mechanisms that the brain uses to compute and process information. So one way he thinks to advance quantitative neuroscience is the interaction between theory and experiments. So using the experiments and the theory to inform each other. So we look really forward to your talk, Antonio. Thank you.
Thank you, Jennifer, and I'm really glad to be here to be able to present today. So I will be talking about this project, Emerging Slow Waves in Premotor Cortex During Task Performance. Brain activity has been associated to that of a critical system, but there is increasing evidence that the global state of the brain can coexist actually with subnetworks residing in different states. So the question we will address here with this work is whether the brain is a critical system, and if yes, which kind of criticality do we encounter here? This project contributes to the HBP goal of investigating the propagation of rhythms during state transitions as a way of studying the neural network. We analyzed in this project recordings from premotor cortex of primate during ketamine anesthesia and the performance of a task. We recorded signals, LFP and spiking activity, the MUA activity of the neurons surrounding the 96 electrodes. During ketamine anesthesia, we observed the very well-known slow wave activity, so the coordinated activation of many neurons in a repetitive way, in a frequency of about 1 Hertz. If we center now the activity on the negative peaks of the LFP, also called NLFPs, we see a sharp increase of firing activity from the majority of the cells. This is the transition from the down state to the up state. There is not only this increase of activity, but this is actually a spatiotemporal patterns of activity that propagates through the network. So as we see here, the activity starts and propagates through the premotor cortex, activating most of the electrodes. Now, during the performance of a delayed reaching task, we also again use the LFP and the MUA signals. And here we see a modulation of the spiking activity due to the associated with the cue and the task, mostly, of course, being premotor cortex during the related to movement. But we also see that observed that during periods where the spiking activity was very low after the movement, so in a kind of resting like situation, there was a high density of NLFPs, of negative peaks of the LFP. So we concentrated in these instances of time. We call them post-movement quiescence. Here now, if we again center on the NLFPs, we surprisingly found a sharp increase of the spiking activity, involving the majority of the neurons recorded. Again, this was in wakefulness during the performance of a task in this period of resting-like activity. This increase of spiking involving the majority of the neurons actually resembled a lot the activity during sleep, and was actually also propagating in the network in a structured way, so as a spatiotemporal pattern. So this, of course, reminded us of the sleep activity and the anesthesia activity. We wanted to understand whether the brain here, the network here, was performing a state transition from the wakeful state to the sleep state. So we analyzed the size of the avalanches, and doing this with the LFP, we saw that this behavior was close to power law, both during the whole task and during the post-movement quiescence. But if we now analyze avalanches of the MUA activity, we see that whereas during the whole task, the activity is not critical, is not power low, during the post-movement quiescence, this is actually close to power low. So first of all, telling us that this is suggesting that we are actually getting close to a critical point, and confirming, as in previous literature, that the LFP is maybe not the good tool to observe criticality in these situations. So with a spiking model of excitatory inhibitory cell connected to each other, we explored the state phase space of adaptation and excitability, setting the network in the slow oscillation phase and in the asynchronous state. But also we move, so to say, the network to go slowly back and forth from the asynchronous state towards the slow oscillation, so reaching the critical transition. And in this situation, we actually recovered in the simulation the sharp increase of spiking activity when close to the critical point. And also this was an activity propagating. So one other thing additionally that we additionally observed is that actually the size of this increase of spiking depended on the previous state of the network. So having a big increase of spiking when the activity previously was low, and having a smaller increase when the activity was high. Of course, we wanted to observe if that was a point also in the data, because this would tell us something about criticality here. And as you may imagine, we actually found the same in data, where we see that big avalanches are actually preceded by a lower spiking activity, and small avalanches from by a higher activity before. This is not what we would expect from classical terms from a critical system, self-organized, like in self-organized criticality, because we wouldn't expect the size of the avalanches actually depend on the previous state of the network. As a conclusion, brain activity is not always critical, but during wakeful quiescent period, this could actually transiently approach a critical point. Here we observe spatial temporal patterns of coordinated activity similar to slow waves during quiet wakefulness. And finally, we propose the log MUA as a good candidate to investigate criticality in the brain. Thank you for your attention. Looking forward for questions.
Thank you very much, Antonio, for that really nice presentation. So we'll encourage everyone to leave their questions in the Q&A, and we'll discuss extensively after 2 more talks. Our next speaker will be Alessandra Camassa, who studied biomedical engineering at the Polytechnic University in Milan. Next, she moved to be a research trainee at the Harvard Medical School in the Division of Sleep and Circadian Disorders. She next pursued a PhD in the Mavi Sanchez Vives lab, which is part of the Systems Neuroscience Group in Barcelona, under the joint supervision of Maurizio Mattia. During her PhD, she studied the spatiotemporal dynamics of the cerebral cortex in unconscious brain states, and during the transitions toward consciousness, focusing on slow wave propagation, cortical complexity, and the network dynamics. Now, as a postdoctoral researcher, her main interest is in computational neuroscience and the study of neural mechanisms that underlie unconscious brain states with a multidisciplinary and multi-scale approach. So thank you, Alessandra. We look forward to your talk.
Thank you very much, Jennifer, for the nice introduction, and thanks for the organizers for inviting me here today. So today I'm going to talk about this project I have been doing during my PhD thesis together with Dr. Sanchez Vives and Maurizio Mattia in the context of collaboration in the Human Brain Project. So this is about the ongoing neural interactions under unconscious brain states. So to understand consciousness is one of the central unsolved problems of today's neuroscience. Why? Well, because to be able to measure consciousness is very important because of the neurological patients where the assessment of the level of consciousness in the clinic is very critical for therapeutic and ethical decisions, but also because we may want to understand the possible emergence of such property also in non-human species or in different biological systems and even in artificial systems. For this reason, it is highly important to identify the brain dynamics and the underlying neural mechanisms that are jointly sufficient for being conscious in a broad sense, so irrespective of the particular contexts of experience. So in the Human Brain Project, there is a work package dedicated to the study of consciousness and the neural dynamics leading to conscious behavior, which is the Consciousness and Cognition work package. But of course, to understand consciousness also means to understand unconsciousness as a ground base for building up conscious behavior. So in this study, we focused on this last point in order to provide an understanding of the neural interactions occurring under unconscious brain states that lead then to the transition towards wakefulness. So we studied here the cortical dynamics under unconscious brain states that may be physiological unconscious states such as deep sleep or slow wave sleep, or induced unconscious states such as different anesthesia levels, where the cortical feedback loops and the thalamocortical feedback connections are disrupted, leading to a dominant persistence of delta oscillations, which are in the range of 1-4 Hz, and slow wave activity below 1 Hz, which are correlated with the lack of behavioral responses. So the activity spontaneously generated by the cerebral cortex under such conditions is known as slow oscillations. So the slow oscillations consist into the alternation of up states, which are active periods of neuronal firing, and down states, which are periods of silence. The up states may be seen as fragments of wakefulness, and they share properties with the awake activity. So what really separates these windows into wakefulness from the actual wakefulness are these silent periods in between, the down states. So all this evidence suggests that we need a detailed understanding of these down states, being these periods critical for cortical function and for the cancellation of conscious processing. So in the current study, we focused on the study of the up and down states dynamics using some novel approaches to understand very well what is happening here. So slow oscillations are a multi-scale phenomenon, and we exploit here the spontaneous emergence of slow oscillations in different preparations as a unifying paradigm to study the cortical spatiotemporal dynamics related to this emerging rhythm in 3 different frameworks. So in vivo and in vitro multi-electrode recordings, where we record the extracellular local field potential activity, and in silico in a simulated multimodular network of spiking neurons. So in this context, we performed a study bridging the characteristic of these bistable dynamics observed in vivo with the one observed in vitro in cortical slices, while exploiting the in silico model to explain the observed phenomena and overcome any possible experimental limitation. So we studied here the network phase and synchronization dynamics during the bistable regime of slow oscillations at multiple scales, but also to capture the multi-scale processes underlying network activity, we studied network synchronization dynamics at 2 different levels. So between neuronal populations in the local field potential, which is made of the neuron's synaptic input current, and then in the multi-unit activity, which is signal made of the output action potentials generated by the neurons. So we computed here the instantaneous phase at each network node or recording electrodes using the analytical signal framework. So this approach entails transforming a real valued time series into a complex phasor, whose modulus and argument in the complex plane represent the signal instantaneous amplitude and phase respectively. So then the instantaneous phase computed, as I explained here, was used to quantify the network level phase synchronization as a function of time estimated by the Kuramoto order parameter. So we found then that the input and output synchronization dynamics bring different information about the network dynamics. So we show here network dynamics through dynamic phase histograms, where the color scale indicates the number of electrodes with a given phase at a given time, and the phase bundles marked by dark pixel over time indicate many sites with the same phase. So then in the middle panel here, we plot the Kuramoto order parameter corresponding to that activity. And in the bottom panel, we plot the corresponding activity for the multi-unit activity and local field potential in our 3 cortical networks. So in vivo, in vitro, and in silico. So the dynamic phase histograms revealed differences in the synchronization dynamic of the multi-unit activity and local field potential, supporting again the idea that these two components of the electrophysiological signal bring different information about network dynamics. So on one hand, we have in the multi-unit activity a pattern of synchronization characterized by a quite regular behavior with a prevalence of in-phase locking during the up states, characterized by high Kuramoto order parameter, and an asynchronous stochastic behavior during the down states in which the Kuramoto order parameter decreases significantly. On the other hand, in the local field potential, we found an intermediate level of synchronization associated with the up state and what we call a metastable dynamics during the down state. So the metastability in the brain is intended as the transient persistence of states which are different than the natural equilibrium state for the network. So in such conditions, the elements of the neural system adjust the mode of interaction in order to generate adaptive behavior within a continuous changing environment. So in this context, the metastability of the down states is characterized by two phases. The first one is a sustained period of high synchronization, which occurs right after the up to down transition, where the collective phase behavior shows an exponential trend and the Kuramoto order parameter is close to 1, so very strong synchronization. Then a second period occurs, which is characterized by a loss in network synchronization, in which the collective phase randomly fluctuates between plus and minus p. So for this reason, we decided to name these stages the deterministic stage and stochastic stage, respectively. And we know that according to the dynamical state of the network, one stage may prevail over the other. So I will go to the conclusions and show you that first of all, we found that by studying both the input and output synchronization dynamics, we obtain information that were not possible to be obtained by the analysis of one component alone. Then we saw that these newly identified periods within the downstate cycle give rise to a detailed up and down state cycle, which is closely connected to the underlying network and cellular mechanisms known for the generation and maintenance of the up and down states and slow oscillations dynamics. Then, very importantly, we see that the metastability of the cortical down states could be related to the drop of cortical complexity, which is typical of the unconscious brain states. And further, the newly proposed stages of the oscillatory cycle that we presented here may be used to predict the responsiveness of the network in different time windows of the down states. And then, thank you so much for your attention. Thank you to the people who worked with me at this study, so Maurizio Mattia, Maria Vittoria Sanchez Vives, and Andrea Galluzzi, and thank you everybody for your attention.
Thank you so much for this delightful talk. I look forward to discussing with questions after. Our next speaker will be Robin Gutzen, who studied physics in Aachen, Germany. He did his PhD in computational neuroscience with Sonja Grun and Michael Denker in Jülich. Since 2018, and will soon finish, congratulations, Robin. He's worked on validation frameworks for neural network simulations. He's currently working on the characterization of propagating activity patterns in the cortex. He has a real passion for data visualization, which we will see in his presentation. He's involved in the development of multiple open source software tools and has the scientific vision that we can accelerate progress in neuroscience by creating and combining common tools, standards, and infrastructure to facilitate more collaborative and easily reproducible research. So you can find him also on GitHub, and we look forward to your presentation, Robin. Thank you.
Great. Thank you, Jennifer, for this nice introduction. And hi also from my side. I'm really glad to be able to show you today our work how to enable the analysis of cortical wave activity across heterogeneous datasets by the means of an adaptable and reusable analysis pipeline. And let me start by giving you some context about the state of the research landscape regarding the analysis of rhythmic brain patterns, and in particular, propagating wave patterns in the regime of 0.1 to 5 Hertz, which are referred to as slow waves. And as you already heard, these slow waves are very characteristic for brain states like sleep and anesthesia, and are persistently observed across many different species, scales, and different methods of measurement, some of which you can see here. And this richness of diverse data brings a great opportunity to investigate this phenomenon from many different viewpoints. And we just saw some very nice examples of such research insights. However, this heterogeneity of the data and their respective analysis brings also the difficulty to compare results or to relate them to each other somehow. So one cause of this lack of comparability is that analysis workflows are usually tailored towards one specific data type or experiment, and therefore can't be reused for different applications. While on the other hand, if we could bring more of these resources together for cross-domain comparisons, it would yield immediate benefits for the integration into broader analyses, to perform meta-studies, as well as to support modeling approaches. So the question that I'd like to address is, how do we get there? And our approach was to first develop a conceptual design of an analysis pipeline that can, in principle, accommodate these needs from different data as well as different analysis methods. And the key feature to enable this kind of flexibility is modularity. So to be able to combine and interface a series of small generic components to create a specific analysis. So using such a combination of components within this pipeline, the idea is to converge the data to a common description level on which then methods can operate while being agnostic about the data's origin. So that the analysis can finally produce comparable results between the heterogeneous data, and also not only being reproducible, but also reusable by others for further applications. When then actually implementing such a reusable pipeline, many of the demands that come up, like how to present the data and the metadata, the workflow management, the provenance tracking, and so on, many of those are already being addressed by various other open source projects on which we can build our implementation. And here you also see some examples. Thus, with this pipeline, we aim to add value by bringing together existing tools and standards and methods without re-implementing the wheel. And as you might notice, this kind of design approach is not really specific to slow wave analysis only, but can in principle be applied to other contexts as well. So however, to come from the concept to our actual realization of this pipeline for slow waves in particular, you see here how we organize this pipeline. So there are 5 sequentially arrangeable stages, starting from general tasks such as the data formatting and the processing, and evolving to more specific stages to identify and finally characterize the waves. And within each stage, there are various methods available as building blocks, which can be individually selected and parameterized via a configuration file. So in the simplest case, you could piece together the desired stages and blocks without any coding, like a choose your own adventure analysis workflow. And then for other more complex cases, due to this inbuilt modularity, the workflow can be expanded and adapted to further applications by just adding or replacing blocks or even entire stages. So to then actually apply this implementation, we used an initial use case of the pipeline to perform a meta study and to analyze 5 open access data sets of in total 60 ECOG and calcium imaging recordings. And we'll just start these GIFs here. So you see here how 2 example data sets of electrical and optical recordings. It was starting from a representation of the raw data and then progressing analogously through the pipeline by processing the data and then detecting the up transition of the local oscillations then grouping them together into wavefronts, and then also adding additional information like the optical flow vector field, which you can also see here, to finally arrive at the final stage where we can apply various characterization measures on the waves, on which we can then build a statistical comparison between the two data or more data sets. For example, here we put against each other data sets of the two different measurement modalities, as well as different anesthetics. And we're still analyzing this large amount of data, but we immediately saw that there's a certain degree of similarity, which really encourages this direct comparison and a closer look at the differences that we do see. For example, we do see, as we would expect, that the finer spatial resolution of the calcium imaging gives us the possibility to detect more complex wave patterns, which are in general less planar as with less spatial resolution. And besides allowing for the comparison between these various data, there are additional payoffs that we do get from this pipeline approach. For example, it also provides a means for method benchmarking. Since this inbuilt modularity, the methods blocks are interchangeable, and we can therefore run the pipeline on different, using different methods on the same data, and therefore compare the impact of the methods with respect to the resulting wave characterization. And here you see one example of this. There are two alternative methods of detecting these local up transitions, and how this in the end then influences the detected wave patterns. And furthermore, the pipeline can also handle simulated data, and therefore can be furthermore used to calibrate and validate models, as it's, for example, done in this collaborating study by Capone et al. So in conclusion, using an adaptable and reusable analysis pipeline approach enables us to not only perform a variety of meta studies in the vast landscape of already available data, models, and methods, but also allows us to more easily reproduce and further build on that work. Or to put it in more general takeaway, promoting a community of tool sharing is, I think, really crucial to provide the basis for effective collaborations across many labs, and will pave the way to tackle big questions in neuroscience and advance our understanding of brain function in a cumulative way. And this, I think, aligns very well with the ideas of the eBrain platform as well, for which we are currently working towards incorporating this workflow as a showcase, and to make it and also other workflows even more available to the community. And of course, I have to thank our many collaborators across Europe, and as well as the Human Brain Project. And you have here a link which points right now to the GitHub repository of the pipeline, but hopefully soon will point to the publication once it is done. And before I forget, I would also like to mention a workshop in June, which will again tackle many of the topics that we heard today, and might be of interest. So the registration is open, and there will also be a link for that in the chat. Thank you all.
Thank you very much, Robin, for this very, very wonderful presentation. So if I can invite all of the speakers back onto the screen, we'll start the Q&A period. All right. So a first question for Dr. Camassa. How might the spiking neural activity look like in people suffering from sleep deprivation or, say, insomnia? Can your work say something about these kinds of pathological brain states?
Yes, sorry, I had trouble starting my video. Okay, here I am.
Perfect.
Yeah, sorry about that. Yes, so about spike in neural activity in people suffering sleep deprivation or insomnia. Okay, I'm not really an expert about this. I never worked with sleep deprivation or insomnia, but as I mean, I can say that of course the fact that one a person doesn't go into sleep, and especially slow wave sleep, will lead to impaired performance in cognitive and memory deficits for sure. And so also I will say that in the electroencephalography will be for sure slowed down, and maybe during the awake state there can be some brain areas that would go into local sleep or sleep-like activity during wakefulness. So this underlines the fact that the slow wave sleep is a very necessary condition for the brain and for our then awake behavior, because it yes, it improves memory consolidation. It also helps in metabolic reconstruction, because of course it's the only moment in which our brain can be turned off for just a while, so like during the down state. So this is my answer. I don't know if someone
that's really nice, and I think this work and the ways that you're addressing this type of question will be useful for developing therapeutics for this kind of problem in the future.
Yeah, exactly. Thank you.
We have a question for Robin. What is the difference between reproducible and reusable analysis in practice?
So reproducible analysis involves steps like having access to both the data and the code, ideally in some version controlled registered manner, that there is a stable and reliable source for it, as well as having the environment, the software environment to execute the code, and therefore the analysis again in exactly the same manner, since even as many of you might know, even changes in Python versions may change quantitative results. And on top of that, making an analysis reusable involves also to basically having an open source license, which allows you to edit the code, and having and building on standardized data and metadata representation, and using standard software tools, file formats, having things like separate separating parameters from actual scripts, and of course also having a good documentation that a new user can actually understand what's going on and ideally change it.
This is very important, yes. And I mean, this kind of work is making such a big difference in the efficiency of science. It's just open sharing and making new work accessible to everyone. So thank you for working on that. I have a question for Antonio. Leonardo Dalla Porta wonders if your model also displays power law neuronal avalanches with a 3/2 exponents as in the data.
Thanks for the question. Yes, to some extent. Of course, it displays different kind of scaling depending on we actually crossed and tried different states. But close to the critical point, we are close to power law. Although I also have to say the network was quite small in order to reproduce the size of the array, of the recording array. So of course with avalanches you would like to have a much bigger, at least in simulation you can have many more orders of magnitudes.
All right, and if anyone finds they would like to discuss more, please type another question in the Q&A box. Here's another question for you, Antonio. What is the difference between propagating waves observed under anesthesia and the propagating waves that you find during quiet wakefulness? Is there some qualitative or quantitative difference that you've seen?
Thanks, Jennifer. They look pretty similar, actually. Of course, during anesthesia, the level of involvement in the firing, I mean, the level of firing rate is higher. It changes more. And of course, in the during anesthesia, you have many repetition of the wave, so you really have this rhythm. In what we found in our data during the resting-like periods is actually what we thought to be just the beginning of a wave of a rhythm. And this we also actually saw could see in the simulation, where if we by being still being close to the critical point and leaving the network alone, actually started the wave, which would get bigger and bigger.
Thank you for this answer, the question and the answer. There are a couple of questions for me that I can answer. So I mentioned I'm trying to link microscopic features with macroscopic features, right? Talk a little bit more about the microscopic features that I'm considering here. So in this particular work, we're considering changes only in the spike frequency adaptation parameter, which are modeling changes in acetylcholine concentration in the brain. And this impinges on potassium currents, in particular the M-type potassium channels and calcium activated potassium channels. However, this is, you know, we've opened Pandora's box. So after this, I think there's a lot of work to be done modeling many, many molecular mechanisms and understanding how you know, bottom up, these mechanisms are regulating the global dynamics, and then also top down, how the global dynamics are then, you know, regulating, enslaving the microscopic processes. So here, only spike frequency adaptation, which is of course involved in the sleep-wake cycle. But as well, there are certain anesthetic agents that impinge on the cholinergic system. Next, we are looking at damage and disconnection of the different regions, which can be, you know, at any scale from microscopic lesion to, you know, to very global, you know, lesions affecting a whole hemisphere or large portions of the brain. But of course, there, for example, Mallory Carlew in Alain Destex's team has come up with a mean field model of Hodgkin-Huxley neurons. So then you'll be able to model many more microscopic events. So I think this is just really the beginning of what I hope will become a new field studying top-down and bottom-up interactions with these multi-scale models. Hopefully that answers the question. And then about yogic meditation, I actually, I have no idea. Of course, I'm aware that there are changes in meditation in many of the frequency bands. They've been hard for me to understand because you can have studies that define the bands of the power spectra in different places. So we're working on methods that are individual, personalizable to individual people, and hopefully I'll be able to answer that question better later. Thank you. There was a great question for Alessandra. In fact, two related questions, so maybe one by one is easier. How is metastability involved in reducing cortical complexity under unconscious brain states?
Okay, this is a very good question. Thank you. So first of all, we think that the impairment of the local causal interactions between cortical areas or elements of the cortical system may cause the drop of cortical complexity, which is related to these unconscious brain states. And in particular, in this case, related to what I presented, the deterministic stage of the down state, which is a period of strong synchronization and hyperpolarization in which the causal interactions are actually disrupted, we think that this could be actually a very important piece to reduce the cortical complexity. Indeed, in previous studies, in a paper from Rosanova et al. In 2018, they showed that under pathological or physiological states of oscillatory activity, the cortex responds to stimulation with a limited activation followed by a strong off period. This off period is the evoked counterpart of what we call the deterministic stage of the down state, because it is actually a strong hyperpolarization period which breaks the causal interaction, causing the drop of cortical complexity. So on one hand, the deterministic stage may be responsible of these mechanisms, and on the other hand, the stochastic stage could be associated with the recovery of the network interactions that are needed to support the activity during the next up state. Yes, so this is my answer.
Great, and I'll do the second related question. What happens to the metastability when the cortical dynamics changes toward the asynchronous dynamics typical of awake state? So very related question.
Okay, great. So of course, if we stay within the bistable regime of oscillation, we still observe metastability. But we saw in our study that this metastability is a state-dependent phenomena. So we experimentally modulating the cortical activity within the bistable regime of oscillation, both in vivo and in vitro with different cortical modulations, we saw that actually modulating the slow wave activity dynamical properties, we obtain also a modulation of the metastability. In particular, what we are changing basically the relationship between these two stages of the downstate and the downstate duration. So in some states may prevail the deterministic stage, and in some others there can be also a greater part of stochastic stages. And so in particular, to answer precisely the question, while moving towards wakefulness or towards, for example, lighter anesthesia states in vivo, we saw that this deterministic stage become shorter. This means the adaptation recovery mechanisms become faster, so the hyperpolarization after an up state becomes recovered faster, and so the rhythm also of the slow oscillation become faster, the frequency of the oscillation become faster. Thank you again for these questions.
Really nice questions. Okay, I think we have time for about one more if we'll stay on time. What a great job you did. All right, so Robin, just wondering, what are some examples of questions or applications this pipeline could address that would otherwise be a lot harder to investigate or not feasible at all?
Right, yeah. So I think a kind of question which is very offers itself to these kinds of generalized pipelines is that it since there are very few setups where different kinds of measurement techniques are used within the same setup or different animal models. So this offers a quite unique opportunity to identify capabilities and also biases within these setups, which would otherwise not really be identifiable. And another kind of question would be, for example, one that I came across in the literature research that I did find multiple papers reporting velocities of waves, which they measured, which would be quite different, but there was no way to see where this difference came from. And actually digging down in there then would require such a an analogous analysis of the different data and setups. And this would be also a way to leverage all these emerging databases of open access data. So there needs to be also basically a big data analysis approach mirroring these big data databases that we get more and more. And I do expect that there will also other applications basically falling out of this design, which we already saw by being able to benchmark methods and quantifying the influence of individual parameters.
Well, this is a very exciting time for research, that we're able to collaborate together and release open tools that we can all, benchmark and ameliorate working together. I think it's really accelerating our progress and our goals, and it's an optimistic time for science and humanity, I think. So thank you to all of the participants so much for your wonderful talks, and for all of the dynamic engagement with the speakers, and to the team who organized this Brain Matters episode. Thank you all for joining. I wish you a great day.